Biofilms are complex communities of microorganisms that stick to surfaces and are surrounded by a slimy, sticky matrix. These communities are made up of bacteria, fungi, and other microbes that live and work together. The image shows a vibrant, green-blue biofilm growing on a smooth surface. It has a slimy, textured appearance with a soft, natural light. Biofilms are important because they allow microbes to thrive and interact in ways they can't as individual cells. Understanding biofilms is crucial as they can have both beneficial and detrimental impacts in areas like medicine, industry, and the environment. Biofilms are complex microbial communities that adhere to surfaces and form a protective matrix. The microbes in a biofilm are encased in a self-produced extracellular polymeric substance, EPS, that binds them together. Biofilms are found in a wide range of environments, from natural habitats to industrial settings and even in the human body. The presence of biofilms can have beneficial or detrimental effects, depending on the context. Biofilms are complex, dynamic communities of microorganisms that form intricate structures. The key structural component is a matrix of extracellular polymeric substances, EPS. EPS is composed of polysaccharides, proteins, lipids, and DNA, which provide structural support and help the biofilm adhere to surfaces. Biofilms are often described as a city of microbes due to the diverse bacterial communities they contain. The specific composition of a biofilm can vary depending on the microbial species present and the environmental conditions. The image on this slide shows the key stages of biofilm formation, from initial bacterial adhesion to the final dispersion of cells. Biofilms start with free-floating planktonic bacteria that attach to a surface. The bacteria then multiply and produce a sticky extracellular matrix, forming a layered structure. As the biofilm matures, it develops a complex 3D architecture with channels for nutrient and waste exchange. Eventually, some bacteria detach from the biofilm and disperse, potentially colonizing new surfaces. Understanding this biofilm life cycle is crucial for managing and controlling biofilm-related issues in various industries and applications. Biofilm formation is a multi-stage process that bacteria undergo to establish a complex, three-dimensional structure. The first stage is initial attachment, where free-floating bacteria attach to a surface. In the next stage, irreversible attachment, the bacteria produce an extracellular matrix that strengthens their grip on the surface. As the bacteria multiply, they form microcolonies, the third stage of biofilm development. Finally, the mature biofilm stage is reached, where the biofilm becomes a highly organized, three-dimensional structure. This sequential process is critical to understanding biofilm behavior and developing effective control strategies. Biofilms are complex communities of microorganisms that adhere to surfaces and are encased in a self-produced extracellular matrix. Nutrient availability is a key factor. Biofilms thrive in environments rich in sugars, proteins, and other nutrients that provide the building blocks for bacterial growth and biofilm development. The surface properties also play a crucial role. Rough, porous, and hydrophobic surfaces offer more favorable attachment sites for bacteria, promoting biofilm formation. Environmental conditions like temperature, pH, and oxygen availability significantly impact biofilm formation by influencing bacterial growth, metabolism, and survival. A higher density of bacteria increases the chances of cell-to-cell interactions and communication, which promotes the formation of a robust biofilm structure. Biofilms are complex communities of microorganisms that thrive in natural environments. They play a vital role in ecosystems, contributing to nutrient cycling, ecological interactions, and even the formation of geological structures. Microbes within biofilms help decompose organic matter, fix nitrogen, and break down pollutants, maintaining the overall balance of the environment. Biofilms also serve as a protected habitat for a diverse range of organisms, supporting their growth and survival. The image on this slide shows a close-up of colorful rock formations covered in algae, highlighting the visual impact of biofilms in natural settings. Biofilms are a significant problem in industrial settings, impacting various industries like food processing, manufacturing, and energy production. They can cause fouling, corrosion, and reduced efficiency in equipment, leading to costly maintenance and downtime. Biofilms can also contaminate products and pose health risks to consumers. Understanding biofilm formation and control strategies is crucial for industries to maintain operational efficiency, product quality, and safety. The image shows a close-up of a pipe with a thick layer of green, brown, and red biofilm, with visible bacteria and other microorganisms, in a dimly lit industrial environment. Biofilms are a major concern in healthcare settings, as they can form on medical devices like catheters, implants, and prosthetics. This can lead to serious infections and complications for patients. Biofilms also contribute to antimicrobial resistance, making these infections harder to treat. Biofilms are often associated with chronic infections, such as wound infections and urinary tract infections. Biofilms in healthcare settings can increase patient morbidity and healthcare costs through healthcare-associated infections, HAIs. It's important to understand the risks posed by biofilms and take steps to prevent and manage them in order to provide the best possible care for patients. Biofilms are complex, protective structures formed by bacteria, making them highly resistant to antibiotics. The biofilm matrix acts as a physical barrier, hindering the penetration of antibiotics to the bacteria within. Bacteria in biofilms exhibit altered gene expression and metabolism, further contributing to antibiotic resistance. Biofilms harbor persister cells, dormant, highly tolerant bacteria that are difficult to eradicate. Biofilms provide an environment that facilitates the spread of antibiotic resistance genes through horizontal gene transfer. Understanding these mechanisms is crucial for developing more effective strategies to combat biofilm-related infections. Biofilms are a major contributor to chronic infections, as they protect bacteria from the immune system and antibiotics. Chronic infections are often persistent and difficult to treat, leading to prolonged illness, discomfort, and increased healthcare costs. Examples of chronic infections associated with biofilms include cystic fibrosis, peritonitis, urinary tract infections, and implant-related infections. Our research is focused on developing new strategies to disrupt biofilm formation, enhance antibiotic penetration, and target biofilm-specific mechanisms in order to improve treatment of these challenging infections. Biofilms are complex microbial communities that require accurate detection and identification for effective management. Various methods are used to study biofilms, including microscopy, culture-based techniques, and molecular analysis. Microscopy, such as confocal laser scanning microscopy, allows us to visualize the detailed structure and architecture of biofilms. Culture-based methods enable us to cultivate and identify the specific bacteria present in the biofilm. Molecular techniques like PCR and next-generation sequencing provide a comprehensive analysis of the microbial community within the biofilm. Biofilms are complex microbial communities that form on surfaces and in various environments. Visualizing these biofilms is key to understanding their structure, composition, and how they develop and change over time. There are a range of imaging techniques we can use to study biofilms in detail, including microscopy, spectroscopy, and other advanced imaging methods. These techniques allow us to see the diverse bacterial communities that make up biofilms, as well as the extracellular matrix that holds the biofilm together. By using these imaging approaches, we can gain valuable insights into the inner workings and dynamics of biofilm formation and behavior. Biofilms are complex communities of microorganisms that can form on surfaces and cause various problems. There are several methods we can use to disrupt and remove biofilms. Mechanical disruption techniques like scrubbing, scraping, and ultrasound physically break up the biofilm structure. Chemical treatments using enzymes, surfactants, and biocides can target and dissolve the biofilm matrix. Laser ablation is a precise method that can remove biofilms without damaging the underlying surface. Choosing the right approach depends on the specific application and the nature of the biofilm. Combining multiple techniques is often most effective for thorough biofilm removal. Biofilms can be a major challenge, as they are complex communities of microorganisms that adhere to surfaces and can be difficult to remove or control. There are several key strategies for managing biofilms, including mechanical removal, chemical disinfection, non-chemical methods, and bioaugmentation. Mechanical removal through scrubbing or brushing can be effective, but the success depends on the specific surface and biofilm properties. Chemical disinfectants and antimicrobials can kill biofilm microbes, but resistance can develop over time, so this approach has limitations. Non-chemical methods like UV radiation, heat, and ultrasound can also be used to damage biofilm structure and inhibit growth. Bioaugmentation, or introducing beneficial microbes, can help outcompete the biofilm-forming organisms and provide a more sustainable control approach. Each of these strategies has strengths and weaknesses, so a combination of approaches is often the most effective way to manage biofilms in different contexts. Surface modification is a key strategy for preventing bacterial adhesion, using materials that resist attachment and incorporating antimicrobial agents into surfaces. Controlling environmental factors like temperature, humidity, and nutrient availability can significantly impact biofilm formation. Maintaining optimal conditions limits bacterial growth and biofilm development. A regular and effective disinfection and cleaning procedures are essential for removing existing biofilms and preventing their formation. Using appropriate disinfectants and cleaning methods is crucial. Developing and employing biofilm inhibitors that target specific mechanisms of biofilm formation can offer a targeted approach to preventing biofilm development. Biofilms can be a significant issue in water treatment plants, impacting efficiency and water quality. Regular cleaning and disinfection are essential to control biofilm growth in these facilities. Chlorination and UV light are commonly used methods to minimize biofilm formation. Biofilms can also develop in water distribution pipes, leading to taste and odor issues, as well as corrosion and pipe blockage. Regular flushing of the distribution network and ongoing water quality monitoring are important to help mitigate these biofilm-related problems. Biofilms in food processing can harbor dangerous pathogens, causing spoilage and foodborne illness, a serious public health and industry risk. Biofilms are difficult to remove from equipment surfaces, requiring specialized cleaning and sanitation procedures. Food safety regulations mandate strict hygiene standards, making biofilm control critical for compliance. Biofilm contamination can result in costly product recalls, production losses, and financial penalties for manufacturers. Biofilms on medical devices can lead to serious infections, causing prolonged hospital stays, increased healthcare costs, and even mortality. It's crucial to effectively manage biofilms on devices to prevent these complications. Choosing materials that are resistant to biofilm formation is critical. Smooth, hydrophobic surfaces with antimicrobial properties are less susceptible to biofilm growth. Various surface treatments can be applied to medical devices to inhibit biofilm formation, such as coatings with antimicrobial agents, hydrogels, and nanomaterials. Thorough sterilization and disinfection protocols are essential to eliminate existing biofilms and prevent new ones from forming. Autoclaving and ethylene oxide gas sterilization are commonly used methods. Biofilms and biofouling are significant problems in a variety of industries and applications. Marine biofouling. Buildup of biofilms on ship hulls increases drag and fuel consumption, requiring regular cleaning and maintenance. Industrial fouling. Biofilms can form in pipes and equipment, causing blockages, corrosion, and reduced efficiency. Water treatment. Biofilm formation in water systems can negatively impact water quality and treatment processes. Biofilms can accelerate corrosion by providing a niche for microorganisms that produce corrosive byproducts. These microbes can utilize metals as electron acceptors, leading to material degradation. Biofilms can create localized environments conducive to pitting corrosion, which can weaken metal and lead to potential failure. Biofilm-induced corrosion is often referred to as microbiologically influenced corrosion, MIC. This type of corrosion can be particularly challenging to control due to the complex interplay between microbial activity and metal degradation. Biofilm-induced corrosion can lead to leaks and spills in pipelines and other infrastructure, impacting ecosystems and human health. Wastewater treatment systems are prone to biofilm formation due to the presence of organic matter and nutrients in the water. Biofilms can form on various surfaces like pipes, filters, and tanks within the treatment system. Biofilms can negatively impact the efficiency of wastewater treatment processes. They can reduce the effectiveness of biological treatment processes and increase the risk of clogging and corrosion. This can lead to poorer removal of pollutants and overall reduced performance of the wastewater treatment plant. Understanding biofilm formation and its impacts is crucial for optimizing wastewater treatment operations. Biofilms are complex microbial communities that can be harnessed for bioremediation. The enzymes produced by biofilms can be extracted and used to break down pollutants. Biofilms enhance degradation by allowing different microbes to work together synergistically. Biofilm communities can be introduced to contaminated sites to boost bioremediation efforts. Biofilms can remove heavy metals through biosorption and bioaccumulation, cleaning up polluted water and soil. Biofilms have a wide range of applications in biotechnology, including bioremediation, biofuel production, biocatalysis, and bioplastics production. In bioremediation, biofilms can be used to clean up contaminated environments by breaking down pollutants like heavy metals, pesticides, and organic contaminants. Biofilms are also used to produce biofuels such as ethanol and biodiesel from renewable resources like algae and waste biomass, providing a sustainable source of energy. Engineered biofilms can be used to produce enzymes and other biocatalysts, which are essential for various industrial processes in food processing, pharmaceuticals, and bioremediation. Biofilms can also be utilized to produce biodegradable plastics from renewable resources, offering an alternative to traditional non-biodegradable plastics that contribute to environmental pollution. Biofilms are complex, interconnected microbial communities that form on surfaces. Scientists are working to design new materials and surfaces that resist biofilm formation by understanding the surface properties that influence attachment. Research is exploring the complex interactions between biofilms and their surrounding environment, including how they influence microbial communities, nutrient cycling, and ecosystem dynamics. New approaches are being explored to disrupt or eliminate biofilms, focusing on targeted therapies that specifically disrupt biofilm structure or metabolism. Biofilms are being harnessed for various biotechnological applications, such as bioremediation, biofuel production, and the development of new biomaterials. Biofilms are complex and diverse, with varying structures and compositions, making them challenging to study and understand. Biofilms formed in lab settings may not accurately reflect their behavior in natural environments or within living organisms, making it difficult to translate findings. Current techniques for detecting and visualizing biofilms have limitations, such as resolution, sensitivity, and accessibility, hindering comprehensive analysis. The host immune system plays a crucial role in biofilm formation and persistence, and understanding these interactions is key for developing effective treatment strategies. Governments like the FDA set standards for food processing to minimize biofilm formation and ensure food safety. Hospitals and clinics have strict protocols to control biofilms on medical devices and surfaces, reducing infection risk. Environmental agencies regulate biofilm control in water systems to maintain water quality and prevent contamination. Biofilm-related issues can lead to legal challenges, especially in cases of contamination or product failures. Biofilm management is a critical challenge, requiring innovative approaches to effectively disrupt and control these complex microbial communities. Advanced technologies like targeted antimicrobial delivery systems and novel biocides can help improve the efficacy of biofilm disruption. Personalized biofilm management, tailored to individual patient needs and specific biofilm characteristics, is key for optimizing treatment outcomes. Integrating big data and artificial intelligence can enhance our ability to predict biofilm formation, optimize management strategies, and deepen our understanding of biofilm dynamics. Exploring biofilm-based applications in areas like bioremediation, biofuel production, and bio-based materials can promote a more sustainable future. Biofilms are complex microbial communities that adhere to surfaces and have significant impacts across various domains. In public health, biofilms can cause chronic infections and contribute to antimicrobial resistance, posing challenges for healthcare and public health. Industrially, biofilms can corrode pipelines, contaminate food, and disrupt various processes, leading to economic losses and environmental pollution. Environmentally, biofilms influence ecosystem dynamics, nutrient cycling, water quality, biodiversity, and ecological stability, with far-reaching consequences. Understanding the importance of biofilms is crucial to addressing these multifaceted challenges and developing effective strategies to mitigate their impacts. Biofilms are found everywhere, They are ubiquitous in nature. Understanding biofilms is crucial as they have major impacts across many domains: healthcare, industry, environment. Controlling biofilm-related infections and optimizing industrial processes that involve biofilms hold great potential for innovation and progress. Developing a comprehensive knowledge of biofilms is key to addressing various challenges we face. The complex, networked nature of biofilms, as illustrated by the image, underscores the importance of studying them in depth. In conclusion, furthering our understanding of biofilms can unlock significant advancements across multiple important fields.